But if there is that homologous relationship, then maybe you're at least predicting a target, even if you didn't have the map that was already in your training set. So I think, that's a question that I don't know if I have an answer to, but there's a lot of issues surrounding evolution that we have to really be aware of to make sure that we don't overfit or overestimate our performance. And I think we've all struggled with them at different times. We've all talked to PhD students who have done things, and then we've had to, very gently show them that what they did is, needs to have the homologs removed, and then the performance goes way down. So that's like a really tough conversation. As far as whether or not evolution can help us predict protein function and structure, I mean, there are definitely many methods where it already does, but I guess we could consider also that evolution has generated lots of related samples, so that for everything that we have that we might try to predict or design, we actually, thanks to evolution, most of the time have many thousands of copies of it with lots of non-trivial perturbations. So I think evolution has already been incorporated into a lot of these workflows already. And so I'm not sure, Li Shen, if that answers your question. If not, maybe bug us a little bit more in the Q&A, because I might have misinterpreted that. But I'd also, the role of evolution, I don't know if Mohammed or Lucy want to talk a little bit about that, but in terms of chemical space or protein space, you have very different considerations. In one case, it's evolution. In the other case, it might be the biases of medicinal chemists. But these are very tricky spaces in the sense that they're very non-random in terms of how they're sampled and what we see. And I'd be interested to discuss that a teeny bit if Lucy and Mohammed, you have some things to say there.
Yeah, no, I think Richard's a really interesting comments, and I'm particularly interested in how we can sort of evolve our own sequences, and sort of leverage the massive directed evolution, or sort of these systems where people are evolving populations of microbes in the lab, and use that to sort of fill in the gaps or understand sort of the real constraints. So these sort of artificial evolution experiments, I think, are really interesting and go hand in hand with that natural data. And particularly trying to sort of understand, how the structural and functional constraints on proteins are reflected in that data, and how we can sort of perturb around them, perhaps to open up new areas of sequence space that haven't been available. There's a sort of amazing work going on at the moment in experimental groups, and figuring out how to sort of analyze and leverage that data is something that I think everyone here has probably grappled with and thought about, and I think there's a lot of potential there still.
Yeah, I do feel like evolution is one of those things which is actually, I think, very much underrated and underutilized in machine learning algorithms, sort of broadly speaking. And I speak, that includes me. I'm always like, I really should make use of evolution, but I don't. And I mean, it's very sort of systematic, right? Because, and actually, I think it's also one of those things which actually could really differentiate machine learning in biology from other fields, because in almost all other fields, you can sort of pretend like your data is IID and get away with it, right? Computer vision, natural language processing, and so on. But I mean, we sort of do the same in computational biology. We pretend that our data is sort of IID. Maybe we do some sort of splits where we're a bit more careful about sequence identity and so on. But clearly, it's a much deeper thing, and I think we would stand to benefit a great deal by actually really leveraging the fact that all things are evolutionarily related. And I'll give two examples. I mean, there's the obvious thing. I was just to say, okay, when you're doing your learning, you're really learning on some sort of underlying graph where things are related by evolutionary distance, right? So maybe you should take that into consideration. But also, I think there are certain inductive biases that come from evolution that are not sort of obvious. For example, the fact that all proteins are evolved probably means that there's some degree of reuse in terms of structural fragments, in terms of even functional pockets, those sorts of things, right? So this is more about reuse as a concept that seems to be a good prior to have about how your data is structured. Another one is the fact that when a new protein evolves, right, it evolves within a pre-existing context of the cellular machinery of all of these other proteins, and essentially sort of presented with this API, right, this sort of interface, literal, like all these molecular interfaces that it has to play with, right? So that's another sort of inductive bias about kind of almost like the generative process through which proteins are, like at least natural proteins, have been evolved. So I feel like this is yet another inexhaustible well of really kind of ideas and pies that we could. And I think we've just barely begun to scratch the surface. And I think it probably has to do with the fact that it requires new machinery that's not developed in the machine in the sort of the broader machine learning world. And so it's sort of harder because we have to do our own inventions much more seriously.
Yeah. And, for any new students who are in the audience, I mean, when we think about the sort of chemicals interacting with proteins interacting with each other, you have the sort of primary data space of a sequence and maybe an alignment of its related sequences. And you can think of that as one of the primary things. You also might think that the three-dimensional structure is another sort of primary data structure. And in between, well, I like to think of in between those spaces, there being some true inverse covariance matrix, some essentially relationship that governs how any variation at one point in the sequence affects the variation at the others. And so in the very early days of evolution meeting structure, we were really obsessed with essentially using the column of the multiple sequence alignment as a feature. And from there, we saw lots of methods where suddenly it was computationally tractable to compute good inverse covariance matrices with some regularization. So all of a sudden, these features were very useful. We could look at the networks of relationships between positions in the sequences when we had very, very large sets of aligned examples, and evolution was sort of powering that core data structure. But that data structure of how the different positions in the sequence vary, which for the largest sequence families we have a very good view on, that is one fundamental way of looking at proteins. Now, of course, each entry, each position, each row in that multiple sequence alignment is a protein and perhaps a different organism. And so understanding the specific interactions, there's the limitation to how useful that can be. But when you think about at least the three-dimensional structure, I mean, that's a very, very useful intermediate sort of structure to think about, that inverse covariance matrix. Now, there are plenty of proteins where there's positive selection and different parts of the multiple sequence alignment are doing different things. So with respect to a given function, that would be a big mistake to think about that data structure. And there's also plenty of problems with transitivity and the fact that you have function and structure interacting and competing for those covariances and inverse covariances. So you shouldn't overinterpret that structure, but it is definitely one of for someone who's more computationally minded, that might be one way to think about something that evolution can give you. And so Deborah Marks has a bunch of papers. There's a bunch of papers from David Jones. There's a lot of various papers that I could dig up and push into the chat if you were interested in that kind of stuff. That's maybe a little more specific than this panel warrants, but for anyone who's sort of getting into this stuff, that might be useful.
Yeah, I mean, just sort of one quick follow-up. I mean, I certainly, protein structure prediction is one of the main drivers of progress in this space. Has been multiple sequence alignments and extracting these kind of covariance matrices, including sort of the work that DeepMind did a couple of years ago, where they essentially kind of use these things to really do very well. And CAS14 is happening next week. We have some indication of what they've done this time from their abstracts, and it seemed like they have actually gone a step further and said, okay, we don't just look at the covariance matrices, but we look at the entire multiple sequence alignment as sort of a raw object, and we can try to extract presumably higher order correlations as well from that object. So it's clearly has played a very significant role in terms of protein structure prediction. I think it's really been the key thing.
But I guess, I mean, sort of following up on that, it's interesting to figure out what's going to happen as we move beyond these sequence alignments, right? I mean, these alignments are generally generated using models that kind of assume that there are no interactions between the columns. Right? And so it sort of seems a little bit odd that we're busy building alignments, assuming there are no interactions in order to extract the interactions, right? Like, I still don't totally understand how that works. It doesn't seem self-consistent. It seems confusing. And, I was really excited to see some of the work presented at this meeting that sort of seemed to be moving beyond reliance on this sort of alignment step. I mean, it's obviously been incredibly rich and valuable. And, when we did that first work, sort of extracting the covariance information from these sequence alignments, we were astonished that it worked with sort of what we thought were really not great alignments. And obviously people have massively improved on it since then. And I think the move to extracting higher order information is super interesting. But yeah, if we could actually really move beyond needing the alignments at all and figure out how to do it, or how to get this information without going through that first step, it would certainly make me more comfortable about the whole process. Obviously also there's limits to what you can align, right? Like we know the HMMs are fundamentally limited in the homology they can recognize. So kind of being able to go beyond that and recognize deeper evolutionary information or evolutionary kind of relationships, I think is really interesting, because there's huge amounts of sequences in the database that we simply can't annotate at the moment because we're restricted by using those models. They're great models. They're very powerful. They're hard to beat, as we found out. But it can be done, and I think obviously it sort of takes a bit of time. But I'm really excited by some of the work that we've seen recently, particularly training over huge sets of sequences and really going beyond that alignment step. I'm curious to know if people have thoughts about how that might progress. It seems difficult. It's such a fundamental object, this alignment. And maybe I'm wrong. Maybe I'm sort of hung up on this thing that isn't really an issue. So Mohammed, Rich, I'm curious to know what you guys think.
Yeah, I think that these sort of self-supervised methods that seem to extract features that have more value than the columns in the multiple sequence alignment, that's definitely the way to go. And I think what we need to do is look at those models and think, what do we not get from those that we get from multiple sequence alignments? And I think one thing that's interesting is you can still take those features and now align sequences and try to look for analogous parts and domain structure and do all the things you used to do. But now, instead of using just the primary sequence, you have these presumably much more sensitive data spaces where the similarities are, essentially much, they're detectable presumably across a wider evolutionary distance. And so it'll be interesting to think through, are there there are elements of, well, one thing, if you have, say, language model extracted features and you pool them and do different things with them, you map them onto graph convolutional neural networks. It's important to remember that if you have a multiple sequence alignment, there are also some very simple things that you can do when it's like, let's say, many versions of a protein in the flu vaccine, in the flu genome, the influenza genome. You can actually date those alignments to the point where they match up with clinical dates. And you can, look at the mutations and actually infer ancestral sequences. And so there's lots of interesting things that you can do. Now, I think when it comes to function prediction, structure prediction, structure function relationships, then the alignment, basically shouldn't be the primary thing, shouldn't be the thing you start with. You're definitely right. But I think it's actually interesting to think through what are the things that that we get from those models that aren't using the columns as features, but they're actually explicit evolutionary models that people use to do things like understand processes of evolution and ancestral sequences and things like that. So, I guess I would agree, but we also probably want to teach students a little bit about, some of those more basic things, just so that we, remind them that once we get an alignment, there are some things that are actually, it's not a feature, it's raw data and a picture of, the output of an evolutionary process with all the ancestors gone, I guess. But yeah, I agree with you that the features we can get from other methods are really blowing away all these other things. I guess I was just mentioning some of the covariance stuff as just kind of an elegant data structure to think about when you're new to evolution for maybe the more machine learning oriented students.
I think this is a great discussion. Very interesting to think about the biases as well as the power of different ways to represent evolutionary data. So I also want to maybe direct to the panelists a question that's related from Kaushik, which is, can people comment a little bit more about, what do you think are the limitations, right? Are we still mostly limited by the resolution of the data that we currently have, right? And maybe a related question is, like, what do you think are some of the most exciting new types of data modalities that could really transform the machine learning models?
I'll take sort of one gander at this in terms of the data modalities. I do think, and kind of come back to the thing I started with earlier about imagine a structure sort of widely available. Well, things like cryo-EM or even cryo electron tomography become really, really interesting. These are modalities which are placing structures within, well, for the cryo-ET case, it's actually placing within sort of its in situ context, within cellular context, right? So suddenly now you don't just have structure sort of detached from reality, but it's very much structure in its native environment. And with the cryo-EM, of course, you're getting a lot richer data, right? You're not just simply getting a single crystal structure to train on, but potentially really an entire ensemble of single particles to train on, right? So at least in this general area of sort of structure / cellular biology, it seems to me like these data modalities are ones that I think are sort of very much ripe for really kind of machine learning to exploit, which I think is not really happened at a sufficient degree yet.
Yeah, I mean, to sort of follow up on that, there is a huge amount of information being generated as these structural techniques get better and better. And also just as we're able to, they become higher throughput and we're able to probe more and more sequences, right? We're less limited by, what nature has handed us. And we're able to sort of really acquire structural information, functional information, huge numbers of sequences in parallel. And I guess I think, in terms of data modality, like the sky is the limit. People are inventing crazy new experiments all the time where you can measure more and more sort of sequences in parallel in numerous different ways and kind of collect holistic information. And I think the challenge of how to integrate all that data is vast, basically, in order to really sort of understand questions or answer questions about biology rather than just about sort of individual molecules or proteins. So Ahmed's point about sort of cryo-EM being able to like put structures in context, I think is really great. We so often think about proteins like in a test tube or in sort of a crystal structure of just that protein, which is not the natural environment at all. And figuring out how to probe proteins in their natural habitat, in situ inside cells, and understand how that affects how they behave, interact with each other, I think is really important. So I guess I'm trying to say that the data generation techniques are evolving faster than I can keep up with them, frankly. And understanding their pros and cons and the sort of the information you can get from each of them is a huge challenge in itself. It's easy to get carried away with machine learning models and kind of not pay attention to that detail, which ends up being really important when you're trying to translate the models through to the real world. So that I think is also very important.
Yeah. Well, one thing to sort of, when we think about a more detailed understanding of biology, one thing that freaks me out is, thinking about just the rate at which we see new functions described, like the growth in just the number of terms in the gene ontology or in any vocabulary that holds function. It's pretty substantial. Some of that's because of curation and because of people working tirelessly to make those ontologies better, but a lot of it's new discovery, right? So, it's hard to, in broad brushstrokes, as you ask there, figure out what those experiments would be, right? So how would we discover microRNA processing in those 30 associated functions before they were discovered in Arabidopsis, and ultimately in worm and ultimately in humans? It'd be really hard to understand how to do that and what that function would even mean before we knew what the cellular context is, right? So what fraction of functions and protein chemical interactions are known or already in our ontologies to learn with in this sort of multitask setting? I think it's actually much less than half, right? Life is pretty diverse, and so most of the chemical reactions that chemists are working so hard to do in labs to synthesize the many drugs we synthesize and the fine chemicals, most of those things probably are done somewhere in the ocean or in healthy soils. Who knows, right? So that's so for me. I think that there's the direction of the experiments that we already have. One of those when it comes to proteins is what things are we sequencing? And so right now there are big efforts like the Earth Biogenome Project and others to essentially sequence organisms that are sort of more broadly balanced across the tree of life. And then I think from our perspective as machine learners or as protein chemists, that's just absolutely brilliant. We're going to see just a lot of things in the database that will teach us a lot about biology. But once we see all those additional genes, how do we identify which ones are clustered in some way that indicates they have a function, but not labeled in some way that indicates they have a new function, right? And so I think actually just the very large expansion in sequencing of a broader swath of biodiversity combined with methods for new instance discovery, which is something people do in image analysis all the time. I think that has a lot of promise. In terms of the more focused, what organism you're studying and sort of drug discovery, I think I think that there's a lot to think about in terms of what associated data types and what data integrations we need, right? So when you do experiments that show you which genes are present in the cell, what if we had detailed metabolomics? What if we had detailed SAR type experiments for subsets of proteins that would tell us, what metabolites are interacting with it, some target gene that we care about. So it really depends on what your ultimate goal is. But I think integrating different experiments is a lot more possible because of machine learning. So integrating microbiome studies with metabolomics is something that's a lot easier to do now with machine learning. Integrating multiple data modalities is definitely something that machine learning can do better today than it could, than 5 years ago. So I think there are a lot of options for thinking about more complex multiple data type experimental designs that the machine learning enables. So I think that might be a little beyond the scope of the question you asked, but that is definitely something that I'm seeing a lot of exciting development in that area. So.
Just actually, I have a few comments on that point. So one thing, I think that's all true. What I do think is there's a potential sort of opportunity with the machine learning is to try to think about how we can integrate these data modalities in a way that is mechanistically kind of faithful, right? That sort of reflects the underlying relationships and structures between those different data modalities, as opposed to say just kind of toss everything into a bucket in a sort of a black box bucket and just say predict me a response variable that I care about. So that is probably a challenge because it requires some degree of domain expertise, right? So it's not something that you can just sort of do in isolation. You really have to understand what these data are doing. But it just seems like one of those things which ultimately, if we were to derive biological insight, we would have to do. And then the other point I was going to say, which is somewhat unrelated, but it's more has to do with the beginning of the question, which is much of the paradigm in sort of the genomics area and the omics area in general is to go and just generate a ton of data, right? And then use that data sort of post hoc to do some sort of analysis to extract some sort of knowledge. But I do think there are a lot of really exciting modalities, particularly like in molecular interactions, and say like things like protein interactions or transcription factor binding, where one is using things that are more like microfluidics type of technologies that they're medium scale. They're not measuring millions of things; they're measuring maybe hundreds of thousands of things, but they're measuring them with very high fidelity. Right? So you're often getting by physical constants from these measurements. And that I think meshes really well with a sort of an active learning loop, where instead of just sort of fetching a whole bunch of data and then doing the machine learning afterwards, you instead incrementally generate the data while learning a model that's proposing which new data to generate, right? And that is also a general area which I feel like has been sort of underinvested in, but seems like a really potentially fruitful direction to go into.
I think that's a really interesting comment. I just want to follow up on that a bit. So the machine learning, the typical deep learning machine learning algorithms are very good at the following kinds of data integration, where you learn some data-driven latent space and you put all of these points in the same sort of embedding space, right? But as you mentioned, I think a lot of the mechanisms and the physics information is actually lost, right? So what do you think are some of the most important physics and biological priors to really try to incorporate into the machine learning algorithms versus just really trying to rely on the data-driven optimization to infer these associations?
Yeah, I mean, for the molecular problems, I think it's fairly natural, right? I mean, things like geometry, or the fact that there's sort of an underlying physical basis where we know a lot of the physics, frankly, for example, in protein folding. So even if we don't force that to be kind of the underlying functional form of whatever we're learning, we could still, for example, regularize toward something that we know is physical. So in those cases, I think it's actually, it's somewhat clear. I think what gets a little bit trickier is when you think of more systems biology questions, right? Where you're trying to incorporate metabolomics data with gene expression data, with those sorts of things, because you sort of pretty quickly get into a situation where you're essentially trying to construct a cellular simulator or something, right? Or something that can really sort of account for all these various pieces. I mean, I think it's a long-term question. I think for me, at least, what I would advocate for is if you have 2 or 3 data modalities that you're trying to integrate, think about ways in which the biology and the physics and the chemistry interrelate, and then think about ways in which, particularly now with differentiable programming, where we can build these bespoke models very easily. How can we encode that information into our model? And that could be as simple as even just the experimental process through which that data is acquired. That could be reflected into the model. And so it's an art form. It's not something I think where it's like a single silver bullet. It's really something that's going to be almost a part of the culture of building the new generation of biological models.
Yeah. Well, I mean, for me, I think, that in addition, I think I agree with everything Mohammed just said. And I think one thing I would just draw attention to is that with, a lot of machine learning methods, you can actually use them to do the boring stuff, like deal with the nuisance variables that differ between experiments or differ between data modalities. And so they don't always have to be the repository for the core physical model. Sometimes they can be essentially a way to keep some of the nuisance parameters and other sort of alignment issues from interfering with revealing that underlying physical model. And in other cases, you can say, I'm going to compute this physical model on a limited manifold, and there are ways of accelerating it. So it doesn't have to necessarily change in any substantive way what you would do with, say, MCMC in the past. Sometimes it's just a way of essentially allowing that sort of thinking to scale. And then the computation is useful because you can do it on a whole genome, or you can do it on a much bigger set of things. So to me, the degree to which it enables new thinking is, I think, in some cases it's scale. In some cases it's also stability of the model. And so I think there's also many cases where you can do things that you just couldn't do before, but it really depends on the problem there. I think for us, again, back to sort of my interests of sort of structure function, one of the things that I've found is that actually many of the machine learning models, although they're more complex, have actually ended up being more interpretable because there's such a nice machinery for class activation mapping, salience mapping, and these sorts of things. And so we often have these models where we think it's capturing the sort of physical structure function essence of that particular function, but we don't have to take the model's word for it. We can actually look in great detail at what the important features are, and actually see with sort of biophysical considerations that in fact this is the active site and the valence of structural residues that scaffold the active site. Things don't always work out so perfectly like that, but in some cases, I think I would sort of agree with what Mohammed said, but then also just sort of add the details that sometimes you don't have to assume that the model's doing that. You can actually sort of go back in and see which features and elements of the model, with a lot of this great new technology for tracing what the model's paying attention to, and you actually do get you do see these really amazing encapsulations of these physical or functional patterns in the features. So I'm really quite hopeful of progress along this question in particular. So
yeah, I mean, thinking about some of the progress we've seen in the physical sciences, where people are really including physical models in their machine learning models, like using it to sort of reshape the architecture of their models, or in order to sort of really improve the ability of the models to extrapolate. It's very tempting to sort of pick up on the question that James asked how we can include sort of biological models or biological priors in the same way to really allow us to sort of extrapolate beyond the data that we have. And Rich mentioned earlier, we have these great models of evolution and these sort of evolutionary processes that are sitting around. And this isn't something I thought about at all, but figuring out how to include that in our machine learning models somehow, or use that to shape our machine learning models, or characterize or sort of bias the relationship between sequences, I think could be really interesting. And I'm sure people are working on this. I haven't seen a great deal of work in that area, so I'd be really interested to learn, if people are actively thinking about how to do that, because it seems challenging to me, frankly. I don't know if anyone else has any ideas, but yeah.
I would add one other class of priors, which I just thought of, which is still in the systems biology context. People have done a lot of work on building kind of really rich dynamical models, either using ODEs or sort of stochastic equations, where you're actually simulating the dynamics of some process like signaling or transcriptional binding. And now, like Lucy was mentioning, we have really great tools to essentially incorporate these simulators within kind of a differentiable framework. So that, I mean, there's maybe like 2 papers to this that I've come across where one really integrates kind of data-driven based learning with sort of these rich biochemical models, essentially. And that to me is also another direction, which I think is under investigated.
Mm-hmm.
Great, that's very interesting. So we're almost at the end of time. So there's one question, maybe we can take that to be the last question from Ali Riza, which is going back to some of these generative models, right? So basically it's asking, to what extent can we trust the output of generative models, and can we start to synthesize these in practice? And maybe I'll just add to that is, we know that machine learning models are quite good at interpolating, but they're not very good at sort of extrapolating to new regions, right? So especially when we think about really new drug compounds, right? So to what extent can you trust the results in those new domains?
I think this is a great question. I certainly found that trusting models that identify new sequences it has been much easier and much more successful than trusting models that identify new molecules. I guess this has to do with how difficult the spaces are to learn. But extrapolating in sequence space is, so far for me anyway, proved more successful. Whereas extrapolating in molecule space, you need a lot of rules to kind of narrow down the proposals that your models make, so rule out the things that are not valid or that kind of can't be made, or all that kind of stuff. And that's my experience so far.
One thing to keep in mind is, we don't have to necessarily use these models in isolation. And so, I think that a lot of the generative models might be really amazing ways of proposing things that we can still double check with other methods, right? And so and I guess there's really not a lot more to say because that's such an open-ended statement, but I think we want to have things. If we had a model that knew about synthesizability, there might still be another model that tells you, oh, well, here you've got a Heck reaction, and you better optimize the conditions if you actually want to have any regioselectivity, and so there, and then after that, you might actually have someone else who just, essentially double checks that they believe your model of how it's docked to the protein, and that also might take SAR data from some high throughput magical experiment, based on mass spec, who knows? So, if you imagine this deployed in a drug company of any size, then I think you have the opportunity to have a few different people competing and fighting it out with different models that have different objectives at different stages in the drug pipeline. So the other thing is, you can imagine, there are a lot of people who have shown that you don't necessarily need all of chemical diversity space to do things in drug discovery. So, something like 90% of all drugs that are approved every year are variations on antibodies, right? And so if that's the case, then do we really need all of chemical diversity space, or do we need some protein domain mimics that are actually small enough to get into cells, but actually substantially bigger than what we currently do. They have modular synthesis. We could have a controlled vocabulary of 70 side chains, and off you go. So there is a new wave in drug discovery that is sort of pushing these sort of protein domain mimics based out of peptidomimetics, and there we could get back into a space where we could very carefully adopt an active learning approach like what Mohammed was suggesting, because we knew we had a controlled vocabulary, and we could sort of design experiments and also design the more expensive computations to map out the cardinal corners of that space. So I think actually, me personally, I'm thinking that we will move towards something where we have modular synthesis and some control, and we have this giant peptidomimetic palette that we use to generate drugs. But I think I'm a little out there. I don't know if anyone agrees with me, but that's kind of my vote for how we go after making new drugs that aren't antibodies.
Great. And any final thoughts on this, Mohammed?
No, I mean, just to sort of follow this point, I mean, I do think like protein design has been an instance of this in a way where, I mean, I think people like the Baker Lab, they've done a really amazing job of essentially figuring out a subset of protein structure space that you could design really well. You can't design arbitrary folds, but at least within that kind of largely helical region, you could design pretty wide set of geometries. So I think there's precedent for that, and I think that's a sensible from a practical perspective, I think that's a sensible way forward.
Okay. Great, we're at the end of the panel. So thank you so much to the panelists. Really interesting discussions. I learned a lot from the process. I'm sure the audience as well. So thank you, Rich, Lucy, Mohammed, for joining us and for sharing your insights.
And thanks for the invitation.
Thank you, and thanks. Enjoy
Mohammed,
the rest of your
Lucy,
day.
great to be on a panel with you.
Yeah, that was good.
You too.
Thanks to the
Bye,
audience.
everyone.
Bye.
Okay, so I think we have one quick spotlight presentation before our final keynotes. I'm Rita. Do you want to share your slides?
Yes. Okay, hi. Thank you to the organizers for giving me a chance to present this work since I had some connectivity issues yesterday. And I'm happy to present this work on behalf of my team at UCSF. The title of this work is Response-Weighted Elastic Net for Prediction in Breast Cancer Cell Lines and Biopsies. And really the purpose of this study was to be able to predict drug resistance and recurrence in breast cancer patients with high risk early stage breast cancer. Our focus was to predict the patients with extreme resistance, so our extreme non-responders. And so we identified pretreatment gene expression features from biopsies in the I-SPY clinical trial, which is an adaptive platform clinical trial where there's early, interregimen, mid-stage, and late endpoints. We're uncovering strong associations of resistance with tumor subtype. And so what we were trying to build is a subtype specific response predictor. And then we had identified MRI features that were predictive of recurrence within 3 years for those patients that had follow-up. We had 990 patients on 7 drugs, and these include immunotherapy agents and other agents. We developed a method leveraging elastic net, and it is a response weight-based method that we use to really focus initially on sensitivity in terms of those drugs on cell lines and then on biopsies. And then for this particular piece of work, we were more focused on, as I said before, the resistant patients. And so this work really evolved from work that was done at the Broad on cancer cell line, a large cancer cell line profiling study, where we took 343 cell lines and 242 cell lines and 353 compounds. Each cell line was treated with a drug, and we were trying to predict chemosensitivity on those cell lines. This work is an interactive resource that was created in identifying cancer genetic and lineage dependencies targeted by small molecules. So we wanted to see how this method would work in patient biopsies. And what we found is that our method achieved higher predictive power on these resistant patients compared to standard elastic net. And we also found that patients that were treated with standard chemotherapy alone, we found two genes, one HIP1R, which is known to target PD-L1 to lysosomal degradation, was strongly associated with extreme resistance, as well as some other immune modulators, which is really interesting because there really aren't good biomarkers for chemotherapy to this day. So the standard of care for these patients, none of the biomarkers have really made it into the clinic. And so we also saw that there were different expression profiles based on subtype. And the subtypes we were looking at were hormone receptor positive, negative, HER2 enriched, and triple negative patients. And finally, when we looked at gene expression and MRI and other clinical features such as demographics, what we found is that our MRI feature actually were our strongest predictors of who was going to recur. And this includes functional tumor volume and tumor volume difference along a patient's treatment journey. And this is all part of the I-SPY 2 trial, which is adaptive, which really means that we can incorporate some of these biomarkers that we're finding through these studies and really integrate it back in to the design of the trial. And also that our method, the response-weighted elastic net, it's pretty robust even with small samples. Since clinical trials, some of our arms or our experimental arms have very, very few patients. And for more information on the method itself, here are two references. And that's it. Thank you.
Great, thank you. Thanks for the spotlight presentation. Okay, great. So I think we have our final keynote speaker, Marinka. Hi, Marinka. Can you see if your slides work?
Yes. Hi, James. Let me share my screen. Great. Can you see my first slide with the title?
Yes,
great.
Looks great. Okay, great. Yeah, so we're very happy to have Professor Marinka Zinnik as our final keynote speaker for MLCB. So Marinka received her PhD in computer science from University of Ljubljana, and then she was a postdoc at Stanford, and now she's an assistant professor in the Department of Biomedical Informatics at Harvard Medical School. So she's been doing a lot of really interesting work in machine learning, especially in graph neural networks, and she'll be telling us about graph neural networks for biomedical data. Looking forward to your presentation, Marinka.
Thank you, James, and thank you, the organizers, for inviting me here today. I'm very excited to talk about graph neural networks and how they have enabled us to tackle some exciting new problems in the area of therapeutics, and more broadly, how they're useful for biomedical data. So the central concept and data representation that we'll be working and discussing in this presentation are networks and graphs. So the way I see networks and graphs are, for me, general language of interconnected complex systems and interconnected data that really allow us to represent a variety of different type of interactions between individuals, within society, or within a given population, all the way down to interactions and associations between different diseases and codependence of those diseases within an individual, and all the way to the level of cellular and molecular interaction networks. And with networks and graphs, we can really span these multiple scales, and we can bring together very rich heterogeneous multimodal data in the broadest sense. So that is very exciting, and that has given rise to quite prominent fields of network biology and network medicine that have grown over the last 2 decades. And the challenge becomes then, once we have such data representations and once you construct these networks, how can we learn and reason over those networks? How can we leverage information on how the variety of different biomedical entities interact with each other? How can we leverage that to make useful predictions, characterizations, and generate variety of products, including compounds, which was discussed in the last panel. And so graph neural networks have emerged as a predominant approach for learning representations and for reasoning over such networks graph models. And they allowed us to tackle a variety of different prediction tasks and problems by extracting patterns from graphs and learning good embedding spaces that can represent graphs in smooth manifolds that can be then decoded and utilized for prediction. And the core concept of graph neural networks is that notion of embeddings or representations, which is really a concept that pervades a variety of different types of different data modalities. But in the context of graphs, what network embeddings or node embeddings in the simplest case, what they represent is they mean the following. So imagine we have a particular graph as our input. So it might be a graph that connects where nodes represent proteins, and proteins are connected based on physical contacts and interactions. And we might also have information about what drugs are targeting proteins and how those drugs co-depend on each other. So that might be an input graph and network that we are analyzing, such as an example shown in the left part of the slide. So the core idea of embeddings is to specify a deep transformation function, here denoted as f, that will take every node from the input graph on the left, and it will map it to some d-dimensional embeddings vector that has a specific desired property. So most often those properties are that we want to capture this topology of the graphs well. So for two nodes U and V that have similar network neighborhoods in the combinatorial graph space, then graph neural networks that are parameterized and represented here by these functions f allow us to also learn embeddings for those two nodes that are positioned close together in the learned embedding spaces. So the core idea is then to optimize the embedding spaces in such a way that they can capture the topology of input graphs. And at the same time, once we have embeddings in these vector representations, we can leverage them for a variety of different problems. So the exciting advances in graph neural networks have now allow us to apply deep learning much more broadly, not only to biomedical images or biological sequences, but now we can apply this powerful paradigm of representation learning to any graph, any network, which is particularly exciting for a variety of different problems in the context of biology and medicine. And this creates many opportunities, some of which I will discuss and present today. So our plan for today is the following. I will cover some of two recent algorithms and methods that we have developed in my lab. In particular, those are methods for learning powerful representations of subgraphs, and I will motivate why that is important and really useful for a variety of different problems in biomedical data. And then I will also address and discuss the problem of how to learn in low data regimes where we have very scarce labeled data. In the context of new algorithms and methods that I will describe, I will also discuss several applications that those methods have enabled in the area of therapeutics, in particular modeling drug treatments and identifying repurposing opportunities for emerging pathogens. Finally, I will conclude this talk with an exciting announcement on Therapeutics Data Commons, which is an initiative that we have recently started at Harvard and aims to bring the area of machine learning closer to machine learning scientists who are developing exciting new models and are really seeking to see how those models can tackle meaningful tasks in the area of therapeutics. Okay, so let's start. So the first part will focus on learning representations for subgraphs that where I will describe a novel algorithm that we have developed and how that algorithm has really enabled us to tackle and to investigate and study drug treatments and identify with high accuracy effective drug treatments. So if I abstract away much of the details, the simplest context our question here will be to be modeling and trying to answer the question of what drug or potential novel compound treats a particular disease. So at the simplest level, the way we can think of it is as we have a large bipartite graph, such as shown here in this slide. We have on the left a large number of drugs, and we have on the right a large number of diseases. And we have information about what drugs are indicated for what diseases, represented by these by black lines here. And so the goal then becomes, how can we leverage that information effectively, and how can we learn some of the drug treatment mechanisms in order to predict what diseases a new drug, a novel drug in development, or a drug that is on the market, but we want to repurpose it, what diseases that drug might treat. So we want to predict these new links in this particular bipartite graph representation. And so our key insight for doing that come from a rich field of pharmacology that often describes drugs and diseases as objects that are, and try to model them as objects that are far more complex than individual nodes in the graphs. And so this is what we will do here as well. So we will represent diseases as subgraphs of rich protein interaction networks that are defined on disease-associated proteins. And we will represent drugs as subgraphs of protein interaction networks defined on proteins that are targeted by the drugs. So in pharmacology, it is known that a drug is likely to have a positive therapeutic effect on the disease if this drug representation is close to the disease representation in this in an abstract pharmacological space. However, as a machine learning researcher, I wonder, it's an obvious question to ask, how can we operationalize this pharmacological space? What does that pharmacological space actually mean? How can we compute over it? So our idea will be that we will use the power of embeddings to learn powerful subgraph embeddings of disease and drugs to operationalize this concept of closeness in pharmacological space. And we will leverage that to predict novel drug-disease relationships. And you might be wondering, well, why subgraphs and why would that be a justified way of doing this? And I will show later in the talk how modeling subgraphs has enabled us most recently to identify 77 drugs and also experimentally verify that they have that they can successfully inhibit viral growth in the context of the new coronavirus, and verify that on monkey and human cells. Interestingly, 76 out of those 77 predicted drugs do not directly bind to any of the proteins that are targeted by SARS-CoV-2. Instead, those drugs rely on network-based actions, and they cannot be identified by alternative non-network-based methods. So this is a very concrete motivation of why one would want to not model drugs as really networks and graphs and model them as subgraphs that are embedded within a large biological network where drugs exert their effects on their direct targets, and then those effects propagate throughout that underlying biological network to alter the molecules that are perturbed by the disease. So that has motivated us greatly to develop new exciting algorithms that allow us to effectively learn and model subgraphs. And a question for that is, well, why subgraphs challenging? And are they challenging at all for current representation learning and graph neural network approaches? And so there are a number of challenges that we need to solve when modeling subgraphs, and three of those are shown here in the slide. So the first challenge is that we really need to when we think of predicting and modeling subgraphs, we need to predict over structures of varying sizes. So subgraphs defined, for example, by, for example, in the context of drugs, might be of varying sizes because different drugs and diseases have a different number of associated proteins and targets. So the question is then how to represent subgraphs that are not simple K-hop neighborhoods. And also how to represent subgraphs that comprise really of multiple disparate components in the biological network that are far away from each other. Furthermore, there are rich connectivity patterns that might exist within subgraphs both internally and externally through interactions with the rest of the graph. And the question is, how can we inject this information faithfully into graph neural network architectures? And finally, subgraphs can be localized, which is shown as an example in the bottom right part of the slide, where all nodes associated with the subgraph reside in one region of the graph, or they might be distributed across multiple local neighborhoods. And as it turns out, a very rigorous network analysis of current drug mechanisms demonstrates how drug targets, while they seem to be enriched in local neighborhoods, they also consist of some disparate components that are distributed across multiple local neighborhoods and resemble more the picture shown here in blue color. So our problem formulation was the following. Our goal was, we want to specify a model that will learn subgraph embeddings such that this model preserves, maximizes the likelihood of preserving subgraph topology in the embedding space. So practically that would mean that if we are given two subgraphs, say Si and Sj, if those two subgraphs Si and J have similar subgraph topology, they should be embedded close together in the embedding space. They should have similar embeddings. So that is not a new concept. This is a concept, of course, that lies at the core of representation learning paradigm. The question really becomes, what does it mean to have similar subgraph topology? And so here the exciting contribution of a new framework that we developed that we called sub-GNN is that we can capture all key properties of subgraph topology. And so what are those properties that sub-GNN can capture? Those properties are describing the internal structure and internal connectivity between nodes that constitute a subgraph, how those internal structure connect to the rest of the network. And then that structure is quantified in the form of three properties, which are neighborhood, structure, and position within the larger picture of the graph. So the way to think about subgraph neural network or sub-GNN is that it has two key parts. So I will very briefly outline those two key parts in the next two slides. But before then, part one is the part that learns or generates subgraph representations in a hierarchical fashion. What that means is that we specify a neural message passing scheme that will propagate neural messages from some anchor patches in the underlying network, and it will propagate those and send neural messages to subgraph components. And those messages will then be aggregated from at the subgraph level, where the subgraph will receive messages from all random anchor patches, and it will aggregate them. That aggregation is parameterized and tunable. In the second part, well, we specify a routing scheme that round those neural messages to three distinct neural channels, which we call position, neighborhood, and structure channels, which are exactly the three channels that capture the distinct aspects of subgraph topology. So very briefly, what these two parts of sub-GNN do is the following, if I unpack this a bit. So in part one, we specify neural message passing scheme. And in order to do that, we need to define what are the messages that the neural messages that will be sent and exchanged between nodes in the graph, and how those will be propagated. So the way we do that is shown in the right part of the slide in one example, when we have when we just show one subgraph, which is shown here in the right, This subgraph has two components. So we might think that we can think of it as a subgraph that would represent one particular disease. And to capture that, to generate its embedding, we specify a number of anchor patches, randomly sampled subgraphs, which are helper subgraphs that are sampled from the underlying graph. And each of those anchor patches then send a neural message to the subgraph component, this red component, all the messages that are received at the subgraph component level are aggregated, and that generates representation or embedding for that particular subgraph. Now, in the second stage, or in the second part, we specify this the exchange of these messages in such a way that we have different type of neural messages depending on the kind of aspect of topology that we want to capture. For example, if you want to learn what is the position of the subgraph within a larger graph, so position what mean the subgraph might be closer to the core of the graph, or it might be closer to the periphery of the graph, then we need to sample those anchor patches in a different way than if you are interested in capturing neighborhood or structure aspect of the topology. And so for each of these three key aspects of topology, we specify a distinct similarity function and sampling function for anchor patches that then allow us to have a separate dedicated channel for propagating neural messages that get aggregated at the final stage, and then also pulled to get an overall embedding of a subgraph. So an overview, the way to think about this is, if I relate to the graphic that I showed earlier in the talk, is that we have a variety, we have this underlying biological network that is overlaid with a variety of subgraphs representing drugs and diseases. And what subGNN does, it specifies this the way messages are being exchanged between subgraphs and the rest of the graph. And it does so in a very flexible way so that it can capture different types of topology and different types of relevant network information, and which are shown here. Okay, so with this outline of the method, we wanted to first test this idea of learning subgraph embeddings in the context of drug and disease representation learning, which was our motivating problem here. So just to refer back to that problem, our goal was to think of our input data as consisting of a large number of disease subgraphs and drug subgraphs, and edges and links between those disease and drug subgraphs indicate known indication and contraindication information. So the goal is then to take these data, learn powerful disease drug subgraph embeddings, and then do link prediction, but at the level of subgraphs, in order to identify and complete these kind of subgraph level bipartite graphs by predicting new edges that are indicative of indications and contraindications for drugs. So in early stages of this research, we teamed up with Spark Translational Research Program at Stanford Medicine, and we used information on what were the drugs that were repurposed within the Spark in the last 2 decades as a ground truth data that allowed us to get in the understanding into failures and successes of the model. And so what we got from them is a long list of drug disease pairs where drugs were repurposed for diseases, meaning that drugs were successfully indicated for a particular disease. And so we asked the model how highly it will rank the actual drug that was repurposed among all the drugs that are on the market in the US. So for example, we can look at row number 4 from the bottom, which says that carvedilol was ranked at position number 9 out of 5,000 distinct drugs for Chagas disease, meaning that the model was confident that carvedilol is 9th most promising drug for Chagas disease. And you can see that the ranks for the list that list of drug disease pairs that I'm showing here are very low, indicating that the model has successfully identified the drug that was later repurposed and placed it at the very top of the predicted list. So since then, we have been asking a variety of different questions. In particular, the question that is perhaps more meaningful and exciting in the area of therapeutics and relates to the question of how can AI algorithms really advance this research area. And so in that light, we ask ourselves, to what extent can we generate and provide predictions that are really predictions about hard cases, about new diseases, and about molecules that are not yet drugs, which are actually those specific out of distribution learning problems where the promise of AI might be the greatest and where we could also help to domain experts the most. So what I'm showing here is a results of one particular experiment that we did, where we took an underlying biological network from a heterogeneous, from a variety of different resources, but primarily drug central from our collaborators from University of New Mexico. And then we asked ourselves, well, how good do current models, including some of the models that I mentioned in this talk, how well do they perform when we randomly split drugs and disease into training, validation, and test set? And you can see that this set of AROC and F1 scores in the far left are very high, indicating that this problem seems to be fairly easy to solve and address by existing algorithms. However, when we construct training, validation, and test set in such a way that we prevent that similar diseases or diseases that are essentially variants of each other, and the same in the context of drug structure, when we prevent similar diseases to appear in the training set and test set, we see how the performance of existing models goes down substantially. Similarly, if we ensure that drugs with similar scaffolds are present only in the training set or only the test set, but not across training and test set, we can really see how performance of models towards predicting indications and contraindications and off-label use of drugs really drops dramatically. So this is an indication that, while it might be that existing models are not really inducing good and meaningful and generalizable biases, inductive biases in during training, and then they really have problems generalizing to diseases and molecules they have never seen before. And so that observation and many others in this line with similar message has led us to think about in a more principled way about how can we make predictions in settings where data labels are very scarce. And that was also motivated by the pandemic this year, where we are tasked with providing predictions for COVID-19 with having no labeled examples of successful treatment of COVID-19. So in the second part of the talk, I will briefly discuss a recent algorithm on meta-learning for graphs. And then I will describe that algorithm also in the context of a particular problem, and that is of modeling emerging pathogens. So when I say emerging pathogens and emerging diseases, what I mean by that is I will refer to the image that we have seen in the beginning of this presentation, but this one is slightly different, and the subtle change is actually quite fundamental. So again, what we see is on the left, we see a large number of drugs, and on the right, we have diseases. And then we also have a new disease that we have not yet seen before, and for which no good treatments yet exist. So in that light, the goal is to predict what drugs might treat COVID-19, which means predicting these black edges between drugs and diseases, but with this caveat that there are no previously known treatment edges that would relate that new disease with the existing drugs. So it's really prediction in the zero-shot learning setting where we have no prior labeled examples of successful treatments for this particular disease. So the results that I will be describing refer to COVID-19 and a major initiative that my group was part of in Boston, and COVID task force also led by Laszlo Barabasi from Northeastern University and Joe Loscalzo from Harvard Medical School. And that is the initiative that was started on March 23rd of this year, where we were tasked with leveraging and putting to use our best AI toolboxes and network toolboxes in order to provide high quality lists of drug repurposing predictions, meaning high quality lists of drugs that would be good, that according to the algorithms might represent a successful are likely to have a therapeutic effect in COVID patients. And so we did that between March 23rd and April 15th, and then we released the predictions and shared them with research community. And then that led to downstream experimental screening and validation on monkey cells and also later on human cells. And the main challenge to putting our AI toolboxes to use in the context of this problem was really the fact that all of a sudden we were tasked with making predictions for this new disease that we have never seen before and for which very little data is available. So the challenge is that as many have probably seen in their project, really truly generalizing to new phenomena is extremely challenging. And so prevailing methods, including prevailing graph neural network methods, require abundant label information. However, labeled examples are often very scarce. And there are many examples of this challenge. I'm referring to the problem of drug repurposing for COVID-19, but there is similar questions arise where we think about novel drugs in development, when we think about modeling rare diseases, about diagnosing hard to diagnose patients. And so what prevailing methods assumes is that our input graphs are these very rich structures with lots of labeled information, shown here as note colors, whereas in reality what happens is we really have rich graphs, but the number of labels that we have for many most exciting problems that we want to study with ML is fairly limited, and these labeled examples are really scarce. So what we have developed was to address this challenge, and to address this challenge of making high quality predictions when labeled data are scarce, we develop a meta-learning algorithm that we call GMeta that can learn from prior experiences and can form adaptive biases to quickly adapt to new tasks. It can really train much better and much more with less data, and it can learn successfully about never before seen phenomena. So the question is, well, how can we design these good meta learners, and how did we do that in the context of GMeta? So in graph neural networks, the key challenge that we need to solve to successfully transfer knowledge from one phenomenon to other is we need to think very deeply about and be very thoughtful about neural message passing scheme. So what that here, I'm just showing one example where we have a number of graphs, just showing two graphs as an example. And so we have this first graph, and then we have perhaps 1,000th graph. And the question is, well, how can we transfer effectively information from this graph to the graph in the bottom of the slide? So what the way this would typically be done is that we would pass and exchange neural messages between edges in the first graph, and then what those messages would do, those messages would propagate those labels and diffuse them along edges of the graph. However, when the number of labels is a small number, that diffusion or propagation process is highly unstable and highly uncertain. So our key idea was that instead of doing propagation across entire graphs, which is also computationally very inefficient, and what current models do, what we can do instead is we can extract local subgraphs that enclose labels, and then we can learn good representations of those local subgraphs and transfer that representational information from one graph to the other graph, to the third graph, and so on. And so the question that arises is, well, what is the value of these local subgraphs that are used as mechanism for transfer learning in GMeta? And so fundamentally, the question is that when we think of what is the source of predictive power in graph neural networks, there are two types of sources or information or key pieces of information that graph neural networks can leverage. And so one piece of information is label information. And graph neural networks propagate labels with the idea that nodes with the same or similar labels are nearby in the graphs. And so that is the type of inductive bias that can be then used to label the rest of the graph that is not yet labeled. The second type, the second source of GNN power is structural equivalence, where the idea is that nodes with the same label will also have similar local graph structure. They will have similar network shape, similar network motifs in their local neighborhoods. And so when labels are scarce, that means that the power of GNN will really not come from label propagation, but we can leverage structural equivalence much more more strongly. And so local subgraphs do exactly that. They capture local graph structure and with an interesting connection to graph kernels, and they can compensate for the power of GNN by focusing on structure rather than on labels when labels are scarce. We have also studied that theoretically, and I highly encourage you to check the papers, but theoretically we were able to show that the influence of a particular node in the graph on a target node for which we want to make a prediction decays exponentially when we go further away from the target node. And so that means that most of the useful predictive information is really resides in the local subgraph neighborhood of the target node that we want to predict. This idea is also has been empirically observed through many applications and of network biology, where many have concluded that most useful information for predicting phenotypes in the context of protein functions actually is in 2 or 3 hop neighborhoods around the target node. So, and GMeta really makes this very explicit and relies on the notion that when labels are sparse and we cannot get really useful information by propagating those labels because of the high uncertainty, then we can leverage more strongly correlations between the structure of local neighborhoods around target node and labels and use that for transfer learning. Okay, so let me show you a bit of a few results, how some of these ideas have helped us to tackle the problem of drug repurposing for COVID-19. So we used our embedding algorithms to embed a large number of drugs and diseases, including COVID-19 disease, which was represented by a subgraph of proteins that are attacked by, that are host proteins identified by viral host proteins that were identified by the Gordon et al. Study from UCSF earlier this year. So that gave us such embedding spaces, and here is just shown one example where every point represents either a drug and a disease. And so within that space, we were also able to position COVID-19. And then we started asking ourselves, what are nearby drugs and nearby diseases in that embedding spaces? And that information allowed us to provide a ranked list of drugs and a ranked for COVID-19. Of course, this can be done in a variety of different ways. So we had a variety of different graph neural network pipelines. Here I'm just showing 4, A1 to A4. And we first, back in beginning of April, we test predictive power of each pipeline by comparing predictions and ranking lists of drugs against clinical trials for COVID-19 in clinicaltrials.gov. So that gave us very promising results, since these GNN-based algorithms have produced really remarkable performance gains over some other diffusion-based or network proximity-based techniques. That being said, however, considering data from clinicaltrials.gov as ground truth information to evaluate performance of the algorithms is really not the right way to do it, since clinical trials would at best capture the assessment of medical community what might work rather than what actually works. Okay, so what we were fortunate to be able to do was that we teamed up with the National Emerging Infectious Disease Laboratories at BU, and NIDL has done for us a large-scale experimental screen of 918 compounds that they screened for their efficacy against the virus, first in vivo E6 monkey cells, and then most recently in human cells. So that study and work has revealed that 37 drugs had strong effect that were on the virus, inhibiting viral growth over a broad range of concentrations, and 40 had weak effect of the virus. Importantly, the ranked list produced by the algorithms had an order of magnitude higher hit rate among top 100 ranked drugs than prior work based on brute force screening. And this really brings me back to the slide that I showed at the beginning of presentation, where I simply talked about why subgraphs and why networks. So out of those 77 drugs, so 40 plus 37 that shown strong or weak effects, 67 of 76 of those drugs successfully reduced viral infection, but they do not really bind directly to any of the proteins that are host proteins of the virus. Instead, they had these indirect effects and could only be discovered really by network-based strategies, and not by docking-based methods for drug repurposing. And we were also very excited that this ranked list produced by the algorithm and the approach that was a final combination of graph neural networks as well as a few other network science techniques yield an order of magnitude improvement, and in the sense of drugs with positive experimental outcome. Those drugs with strong and weak outcomes were much more strongly enriched at the top of the ranked list produced by the algorithms than what could be possible without meaningful data-driven ranking. So what were some two key machine learning lessons for me and my group that I can share here are the following. So first, by doing this and by participating in this task force and working very closely together with domain experts, essentially having daily meetings when we had discussed the progress, discussed current predictions, discussed results, data sets, and so on. What was really important observation was the domain scientists, in this case, virologists, experimental biologists, and medical researchers, they need to interact with our AI systems, and we need to design them in such a way that these algorithms are able to provide feedback for the machine learning loop. So very early on, we realized that it is not simply enough to provide these kind of simple ranked lists of drugs and their predicted probabilities of their being effective, but we need to provide underlying mechanisms that drive the models to make those predictions. And we were lucky enough that we have previously developed some of the explainability algorithms that we put into use here. And second is this observation that generalizing to new diseases, or similarly, I would argue generalizing to new compounds, is extremely hard, especially when that disease does not really is very different from what we have seen before. It's not a simple variant or variation of what has been seen before. Okay, so to conclude in the last few minutes, I will present a particular new exciting project that to me is exciting because of its high potential in impact to help advance machine learning research and work in the area of therapeutics. And so the last year or so, I became extremely excited about therapeutics. I think they're one of most exciting areas for computational machine learning scientists. However, by talking to many fellow computer scientists, I I often hear that retrieving, curating, processing data sets is extremely time consuming, and more importantly, it requires extensive domain expertise that many computational scientists don't necessarily have. And those data sets are also scattered around biorepositories. There's no centralized data repository for a variety of therapeutics or types of problems. And because of that, many potentially impactful, important tasks remain underexplored in AI ML community because there are no clear data sets and there are no clear tasks that would be meaningful that are defined in this space. So what we have been developing last few months in my group is something we call Therapeutics Data Commons. So Therapeutic Data Commons is an open source data hub for machine learning data sets for therapeutics. It covers a wide range of data tasks from prediction tasks from target discovery, activity screening, efficacy, safety, manufacturing, generative models, covers a wide range of biological products, including small molecules, antibodies, vaccines, mRNAs, and so on. It has numerous data functions, and it's very easy to use. In 3 lines of code, one can get an interesting data set, meaningful data split, and a task that can be tackled here. So our vision for TDC is that it's really a data hub that sits in between domain scientists and machine learning scientists, where machine learning scientists can design powerful ML model, whereas domain scientists identify meaningful tasks and provide relevant domain expertise. And altogether, then this data hub can help us advance algorithms for key tasks in therapeutics. The way to think about its structure is that we have in TDC 3 types of problems. So we support data sets and tasks that can be represented in one of the 3 categories. So the first category is single instance prediction. These are prediction problems where the goal is to predict a property about a single instance, such as a single compound or a single disease or a single protein. The multi-instance prediction problems are those that revolve around predicting properties of multiple instances or group of instances. For example, predicting efficacy of a drug combination. And finally, third group of problems are generative problems, where the goal is to synthesize a particular compound with specific properties. For each of these 3 problems, TDC has a number of therapeutic tasks, and then for each of those tasks, it provides a number of data sets. So we cover a wide range of prediction tasks, and altogether in this initial release, which we just announced last week at the symposium at Harvard, we have 62 data sets that are ready for everyone here to use and explore. I'm just showing here one example of a drug response prediction data set and drug synergy data set that allow us to model and evaluate the power of algorithms for predicting drug synergy scores and drug response, as well as another example for a variety of problems related to biologics. Interestingly, TDC has a number of functions that can support your research in the form of loading the data, performing various types of model evaluations, implement a variety of meaningful data splits, not only random splits, but scaffold splits, for example, for molecules and so on, a number of data processing helpers, a number of molecule generation oracles. I know that in the previous panel, there have been lots of discussion about how can we generate molecules and compounds in a meaningful way. And so TDC provides a number of oracles that can help evaluate and also motivate the development of these generative models in this space. So I will stop here. You're invited to join Therapeutics Data Commons. This is really an open source community effort. We just started it. We have a GitHub repository, a website with all 62 data sets, also Python P package that allows you to get any of the data sets in just 3 lines of code. And I want to acknowledge 4 amazing students from my group, as well as from collaborators at MIT, CMU, and Georgia Tech that are part of this effort. And all papers, code, data that I mentioned today are on our lab website. Feel free to send me emails. And this is our lab website, since our lab is so young that we actually have not yet had a chance to take an in-person lab photo. So I'll stop here, and I can take any question now.
Great, thanks Marinka for the great talk. So we have time for a few questions. In the Q&A, so Adrian has a question about, I guess, for earlier in the talk, why is it that the performance of the off-label usage does not degrade very much?
Okay, yeah, so that's a great question, Adrian. So the reason why the performance for off-label use did not degrade much was because of some of the data sources that we were including in the underlying biological network, where those data sources on the off-label use contained information about or contained information from FDA Orange Book, and that information had data that correlated strongly with the off-label use of drugs. So that was the reason why the performance did not degrade much.
Great. Tonkin also has a question. So the question is, have you considered possible ways of representation learning on heterogeneous graphs? So for example, knowledge graphs, when you have different types of biological entities?
Yes, absolutely. So much of the research that we are doing that we do today goes well beyond protein interaction networks or physical interaction networks as the sole type of edges in the graph. So in the context of drug repurposing for COVID, the final data representation that we used to make predictions was a heterogeneous network of proteins, protein nodes, drug nodes, disease nodes, as well as a variety of different site information and features, including information on symptoms and of diseases and drug structure. So certainly, I think knowledge graphs are an effective and exciting way to represent these heterogeneous sources of information. That, of course, creates a question of how to specify this neural message passing scheme in a thoughtful way, rather than simply think of it as a kind of kitchen sink approach. So we are very much working on projects that involve knowledge graphs in this way.
Great. Maybe one final question. So for COVID, I guess there's growing data from medical records and others of the efficacies of different types of treatment, right, on COVID. So have you considered incorporating those kinds of data for your meta-learning problem?
Yes, so that's a great question. So when we did the initial study in April, at that time there was very little information on electronic health records, and the data that came from electronic health record was extremely messy because of a variety of different challenges, for example, with just coding COVID-related symptoms or coding COVID-related diseases in electronic health records. So we relied primarily on molecular interaction data, the fantastic work that came out of Nevan Krogan lab at UCSF on identifying host proteins of novel coronavirus to produce initial ranked lists of drugs. So since then, we have several projects where we are looking at electronic health record data for patients, as well as looking at combinations of drugs rather than individual drugs, which is what we did in the study that I described here.
Okay, great. Thank you, Marinka, for the great presentation. So this wraps up the Machine Learning and Computational Biology Conference. So thank you, everyone, for all of your participation. Thanks to the speakers and to the attendees and to the poster presenters. And look forward to meeting all of you in person, hopefully next year. And also want to thank Deep Genomics, who provided some of the funding support to support the poster sessions. Okay, great. Well, very nice to meet all of you in the last 2 days, and enjoy your Thanksgiving break.